on an evening like this, when we introduce a new professor of Rhodes University, we introduce her fully. So I have a distinct honor of presenting to you one of our newest additions to the illustrious community of scholars who hold the position of full professor of Rhodes University, Özlem Tastan Bishop. Özlem was born and grew up in Istanbul, in Turkey. As a child, her father often took her to the bank where he worked. So she was exposed to large numbers even before starting school. And this is where her love of mathematics started. Her mathematics marks were always top in her class, and she shared her knowledge of the subject, on the subject by teachers with her classmates. After passing 2 stages of university entry exams, Professor Tastan Bishop was accepted into the physics department at Zeki University in Istanbul, one of the top institutions in Turkey. The department was known to be tough, and the graduation numbers were generally low. While at university, she tutored struggling high school students in mathematics and physics. As a student, she had to take an extra semester, and she chose a biology module. The tiny and crowded world of cells fascinated her, and she decided to specialize in the subject instead of physics. She completed her MSc at the university's Molecular Biology and Genetics Department in 1996. Her first trip out of Turkey took place after obtaining her MSc degree, where she went to the Medical Research Council in Oxford on a British Council's fellowship. This was just one of many fellowships she received. Others included a Turkish government education scholarship, a Monbusho Research Student Scholarship from the Japanese Ministry of Education, a Deutsche Forschungsgemeinschaft, an NRF freestanding postdoctoral fellowship, and a Claude Lyon Foundation postdoctoral fellowship. Clearly, Professor Tastan Bishop followed her passion for science in different countries, cultures, and education systems. In 2003, she received her PhD from the Max Planck Institute for Molecular Genetics and Free University in Berlin, Germany. She did her postdoctoral studies at the University of Texas, University of the Western Cape, University of Cape Town, and the University of Pretoria. After her time in Pretoria, Professor Tastan Bishop joined us at Rhodes University. Since joining us in 2009, she established the bioinformatics discipline, along with MSc and PhD programs in bioinformatics. She has graduated 25 PhDs and 38 master's students and mentored 8 postdoctoral fellows. Over the years, Professor Tastan Bishop has received several awards, including the SASBMB 2005 Prize for the Best Oral Presentation given by Experimental Biology Group, and the South African Society for Bioinformatics Silver Award for 2022. At our university, she has received numerous accolades, including our Academic Merit Awards for 2012, 2015, and 2020, and the Individual Internationalization Award for 2018. Professor Tastan Bishop is one of the founders and the first president of the South African Society for Bioinformatics, SASB. In recognition of her outstanding contributions to the bioinformatics field, she also received the South African Society for Bioinformatics Silver Award in 2022. Her research is well acknowledged nationally and internationally, and she's an NRF-rated scientist. Demonstrating her continuous excellence in her field, she has received numerous grants from well-known international bodies, including the Bill and Melinda Gates Foundation, Malaria Medicine Venture, African Academy of Sciences, Wellcome Trust, and the National Institute of Health. And the National Institute of Health Common Fund, as well as national bodies such as the NRF and the South African Medical Research Council. Last year, Professor Tastan Bishop was selected to represent Rhodes University in the health sciences of the Arua Guild Initiative, which led to her being part of 2 clusters of excellence that were established earlier this year. She serves on the editorial board for Frontiers in Molecular Biosciences and Frontiers in Applied Mathematics and Statistics, Biological Modeling and Simulation section. She is an advisory board member of the F1000 Research Bioinformatics Gateway. She has reviewed numerous articles for many highly recognized international journals, including several ACS journals, Nature Communications, and Wire's Computational Molecular Science. She has published 97 research articles in high-impact journals. She is the editor of the book titled Bioinformatics and Data Analysis in Microbiology. As a scientist in the short term, Professor Tastan Bishop says she would like to impact drug discovery by establishing further approaches and tools to circumvent mutation-related effects in the context of drug resistance and drug toxicity and efficacy. In the long term, she would like to be one of the pioneers in establishing Afrocentric computational drug discovery research, specifically targeted at improvements in scientific competencies that can lead to better health care and stimulate economic growth on the African continent. Now, honored guests, colleagues, and friends, it is my singular honor to invite Professor Tastan Bishop to present her professorial inaugural lecture titled From Istanbul to Grahamstown, Makhanda, Different Countries, Cultures, and Education, but Always Science. Professor Tastan Bishop.
Thank you, Professor Mobizela, this kind introduction. Vice Chancellor, Deputy Vice Chancellors, Deans, Deputy Deans, colleagues, students, friends, and family. Good evening. I would like to welcome you and also thank you for being here and not letting me alone this evening, attending this lecture physically or via Zoom. Okay, I will remember to share my screen now. Okay, this evening I would like to talk about my journey, educational and personal journey. I believe that my journey over the years made me what I am currently as an educator, as a scientist and academic. And I think the title summarizes what I would like to tell you. But before starting talking about these things, I would like to do something else and tell something else. You might know from my previous lectures that I actually like to refer my talks to movies or fictional characters. With the same way, I decided to look at fictional professor characters. There are lots of doctor characters and mostly evil, but something happens when you become professor. Obviously, professors are socially inept or mad or clumsy. Few of them likes action in their life. I could only find one professor, evil professor, Professor Moriarty from Cambridge University, and he's mathematician. And it's very sad to also see that there are not many female professor characters. There's only one that I can recall. She's law professor in the series How to Get Away with Murder. This needs to change, right? But let's see how it goes in real life. Let me tell you a little bit about my journey, my adventures. I would like to dedicate this talk to my mom, my late father, and my brother. And as you can easily guess, that little one is me. And you can follow me from now on with this yellow arrow. I was born and grew up in Istanbul. Istanbul is extremely big city, very crowded, 16 million people, which makes it roughly 114 times more crowded than Grahamstown. It's also a very old city. There is so much history everywhere. But for us as academics and students, I think the most important history related to Istanbul is coffee. Believe or not, the first coffee shop, cafe in the world was established in Istanbul. And also how we brew the coffee currently by roasting and grinding is coming from Ottoman Empire. Why I'm telling this? Because I think many of us cannot end the day without at least a cup of coffee. So it is related to us. Okay, Istanbul is really very big. When I started going university, Boğaziçi University, also called Bosphorus University, because it is located in Bosphorus, I had to take 3 hours to be able to reach the university by 3 buses. And it was the same 3 hours to go back to home. So this was my preparation year when I learned English, as the education system in the university was in English. But then it had to change because it was like 6 hours traveling was too much. And I decided to stay in dormitories starting from year 1 physics, and you call it residences here. And staying in dormitory changed my life a lot. But first, life in physics department, how was it? Physics was fun, but it was very tough, as Professor Vizela told already. So physics department every year took 50 students to BSc, but the graduation level was very low, 5 to 7. At the end of first year, most of my classmates left the university or moved to another department, and it was exactly the same in the second year too. But I was in dormitory. I got very nice friend, Nurgün, and she was from molecular biology and genetics department, talking about these amazing things, genes and genomes and chromosomes that I never heard before. It was so exciting, and I started to take some modules, elective modules from biology. But then I was in between physics and molecular biology. Physics was extremely stressful, but biology, I was not sure if it was really my thing. So I talked to a number of people. One of them was the head of the biology department. She was extremely nice person, Prof. Betül. And she told me that I must get out of the university and go and talk to scientists in a research institute that she worked previously. And she basically arranged a trip to institute called Çekmece Nuclear Research and Training Center. And this was that time in the middle of nowhere. There was no transport during the day, or very little. And I had to take a garbage truck to arrive at the institute. My son loves this story and asks all sorts of about the garbage truck. And now I talked to so many people there, and it really helped me to decide how to go on my way. And one of the physicists there, and there were biologists as well, he told me that you can learn biology anytime, but you cannot do the same for physics. Okay. So I continued with physics department. Okay. And as you can see, we were 6 graduates that year. During that period of time, I also took some modules from electric and electronic engineering department from Prof. Yağmur, and she actually became one of my very first role models. She was young, smart, and very kind. I really enjoyed her lectures and learned about modeling nonlinear dynamics and biological systems. Then, of course, I moved to biology. Okay, I did my MSc in biology under the supervision of Prof. Nazlı Başak. And my actually thesis was about alpha thalassemia. So alpha thalassemia is a blood disorder, inherited one, and we have hemoglobins in our red cells and red blood cells, and they capture the oxygen and carry it to the tissue cells. So each hemoglobin has 2 alpha globin and 2 beta globin proteins, and the mutations on these proteins can cause thalassemia. So my thesis was about establishing PCR methods for detecting the mutations. You might think that this is such a routine thing now, but that time in 1993, when I started my MSc, actually the person, Kerry Mullins, got Nobel Prize the same year establishing PCR. So it was something very big. I also specifically wanted to show this page here to the students. As you can see, it's a photograph copy. They stick to the thesis, and this is how we would do the things that time. Technology was not well established, but it doesn't mean that I'm old, actually. I had many take-home messages from my MSc. I actually underestimated learning biology. Okay, biology was not easy to learn, and it was very different than learning physics. But what helped me finishing physics, I learned how to look at the logics and reasoning behind the biological events. So I cannot memorize. So it was something good to finish the physics first. My next trip was to MRC. And this was my very first international trip. I really enjoyed. I was so privileged to work with Dr. Douglas Hicks. He's a very well-known hematologist. I worked in another disease, inherited disease. This was rare disease, ATRX syndrome. In this disease, alpha thalassemia and mental retardation occurs together. So this was also very interesting because we had alpha thalassemia, but it was not caused by the mutations of alpha globin gene, but another gene, actually ATRX gene or protein, was affecting the expression of alpha globins. So I started to learn more and more about human diseases and mutations by then. And it was my very first publication that came from this study. I learned lots of things there. I think I decided to be scientist after visiting MRC. It was a wonderful place to be, especially after MSc level. I could meet world famous scientists on daily basis and listen to them. I also learned that I could work very hard and visit all the pubs in Oxford. I knew all of them. I worked with French, Canadian, English person there, and it was so hard to understand his accent, but I improved and they also eventually understood my English. But most importantly, I realized that resources are so important for science. And now looking back and looking at the science done in Africa, I actually congratulate the people working in Africa and doing such a wonderful job with such a limited resources. They let me do independent research there, and I learned to be, actually, I realized that independent thinking brings lots of creativity, and this is exactly what I'm trying to do in my group now, and encouraging all my PhD students not to be dependent on me. Okay. My PhD actually started in Japan, University of Tokyo. I got Mombusho Fellowship, and I started to work under Professor Kimitsuna Watanabe. Kimitsuna Watanabe's group was working on ribosomes. There were 3 groups under him. Each of them were doing something different, but related to ribosomes. Ribosomes are these giant machineries with lots of proteins, 50 to 60 proteins altogether, depending on the organism. And 3 ribosomal RNAs come together, make this machinery. And they actually read the genetic code, and tRNAs bind to this machinery and carrying individual amino acids and attach to each other, make the protein. Okay, here you see all this small subunit and large subunit of the ribosomes, the purple ones or all the proteins, and the yellow ones or brown ones are the RNAs. So there was one doctor, Dr. Nitta, we called him Nitta-san. Nitta-san was doing something so interesting. He was working on showing the minimum part of the ribosome which can do the protein synthesis. This was something extremely important. And he was in his experiment showing that actually this is for 1 amino acid and 23 S ribosomal RNA, which is this one all alone, can attach to amino acids. And he was planning to write science article. So it was wonderful opportunity for me to be part of his group. And I did. I started doing the experiments. Unfortunately, nothing worked in my hands. It was disaster. Months and months, nothing just worked in my hands. And Nitta-san politely told me that perhaps I should design my own PhD project. Imagine as a PhD student in 1 year, from dreaming from science article to be in the position of designing your own project. And I did. I was staff thanks to a physics department. And I also set up collaboration with a group in Berlin. Prof. Knut Nierhaus was well-known ribosomologist. And when I arrived Germany, actually the science article was published. And now there is quite difference between German culture and Japanese culture. Japanese culture is extremely quiet culture. And German culture is totally opposite. They are very direct, like Turkish people. And they can really, they like to discuss and argue. And they challenged me from the first day that I arrived about this article. I couldn't tell them that I couldn't reproduce, I couldn't get any of those results. It took 2 months for me to have my courage to go to Knut and ask him to give me resources to repeat all these experiments that didn't work in my hands and do all the controls that I was not allowed to do in Japan. Knut was very kind. He gave all the resources and also a PhD student to me. Ulrich and I started working and it took a month separately, individually. We did the experiments and we had roughly 80 pages of report providing evidence that the calculations in science article was wrong. Now it was my job to Knut said that he have to go back and tell Professor Watanabe and Dr. Nita that they have to write erratum and we can write another science article with what you have actually. So I do not know which one was the most scariest, flying back to Japan with Aeroflot, which was very scary, or challenging a Japanese professor that time as a PhD student. But I did go back. I tried to explain, but they didn't write erratum. And another group in between published, sorry, I want to take this off, actually off, and published another article showing that everything was not correct in that science article. They retracted the article at the end, but meantime, Knut offered me another position and I moved back to Berlin. But Professor Watanabe was so kind. He really kept my registration a couple of months more until he was sure that I was fine in Berlin. So my take-home messages, I had lots of take-home messages and I tell this to my students. I don't need to tell my story to everybody all the time, but I tell them that they shouldn't imagine the results. They should really look at the results and they do every control. And most importantly, if they did wrong, they should accept and come back and repeat it. And they do that. And I feel so relieved when they do that. That's very good. Okay, this was the sentence I heard so many times from Knut. There is no stupid question. And that actually made me learn so many things, because if you are in an environment that you know that you can discuss anything, you can ask anything, you can actually learn quite a bit. Knut also liked to bet. And he on all sorts of scientific questions. I got lots of moncheries from him, but I also lost quite a bit. So it was so it was good. Over the years, I learned a lot from him, but I think I also managed to teach some stuff to him and the group. As you can see, he was quite talented, unlike Ulrich. Okay, but overall, he was really a very good scientist. He was very meticulous, always excited, asking lots of questions, pushing us to our best. And that was quite a bit of learning curve for me. And he was the first person actually identify the third binding site on ribosomes. If you look at the old textbooks, there are only 2 binding sites. And he showed that, but he got quite a bit of difficulty from the ribosomologist. The community didn't want to believe him very long time, actually, even though he collaborated with Johann Frank in certain point and did cryo-electron microscopy, which was showing the third side, but people just was resistant. And because the cryo-EM was new and the resolution was not very good. And my PhD project was even more challenging. I was looking at how these tRNAs would move on the ribosome, including the third binding site, which was not accepted by the community. Over the years, actually, people really work hard to get the crystallography. It took 30 years to get ribosomes crystals. And at the end, these three people got Nobel Prize for that. And other Jonat is started in Max Planck Institute initially. So I was so much exposed to structural biology techniques and learn about importance of looking at the proteins structure and relate to function. So at the end, it was accepted by everybody that Knut was right, that there are three binding sites on ribosomes. We lost Knut on his 75th birthday, actually, in 2016. Take-home messages. I have so many things that I could say, but I was just called to this. And that is my PhD, basically. Postgraduate study is not simply a matter of coming to know, it is also a matter of coming to be. And it is what I feel about my PhD. I did 3 postdocs, and in each postdoc, I learned one structural biology techniques, but I would like to talk specifically about the one in Pretoria, and because this is the time when I started to set up my group, actually. And for that, I'm very thankful to Prof. Joubert. I was postdoc under him, and it was not common thing to apply a grant as a postdoc, but he really supported me, and I got my very first big grant from National Bioinformatics Network. And I had, these are my very first MSc students, Matthias, Fortunate, and Ansi. I also, because of this grant, established set up 3 workshops and with international speakers. And I started to get some phone calls from a professor, Professor Greg Bleich from a university that I never heard before, Rhodes University. And every time he was asking me, can I send more than one student? And I was like, yes, of course, please send. And eventually he headhunted me, and I became senior lecturer in this university to establish postgraduate studies in bioinformatics. Over the years, I worked with amazing, very nice leadership, actually. I color coded according to the position. Okay, so I specifically would like to, I'm really grateful to all of them, but I would like to mention 3 names here, Prof. Joe Dames and Prof. Rick Bernard. Helped me a lot to things going when I started as a senior lecturer here and to establish the things. And I was very privileged to work with Prof. Peter Clayton, roughly 12 years as during his DVC position. And I'm thankful to all of them. I also would like to acknowledge 4 academics, colleagues here. I wouldn't be able to run the MSc program without their support. Since 2011, we are keep teaching and doing what the best that we know, and with their help. Okay, we started with basically cleaning the storage place. This was really how it happened. Sharon and I had to move everything out, dust out. University gave me 50,000 rand. So this was the money that I had to use to establish the lab, basically. So I couldn't, of course, get an architect or anything. I designed the things, measured, and we found people to, carpenters to do the job, carpet, and it was there. And being Turkish helps, I bargained with everybody to decrease the price. Okay, so this was our opening invitation. Over the years, I work with many very brilliant people, actually. Kunut would say young stars. So each of them were amazing. And I really hope that when they leave the group, they feel the same way as I felt when I did my PhD. And I'm hoping that I'm actually putting more than to their life, more than just knowledge, but also helping them shaping their personality and future in a positive way. What do we do as a group? We work on structural bioinformatics and its applications to early drug discovery related projects. And while doing that, we try to introduce unorthodox approaches and tools because there are actually very many gaps in the computational drug discovery. Let me just define structural bioinformatics very quickly. It focuses on the identification and analysis of structures, dynamics, and interaction of biological micromolecules in 3D space. I, by purpose, presented proteins here in three different presentations: cartoon, surface, and coarse grain, to familiarize you that there are many different ways to present the proteins, not to confuse you, actually. Okay, so knowing 3D structures of macromolecules is very important in understanding many biological phenomena. I tried to explain a little bit by ribosomes and 3D tRNA binding sites, but also it is very important for drug discovery. We call it rational drug discovery if structures are used. And there are many FDA-approved drugs actually in the market designed by using the structures of proteins, and HIV/AIDS drugs are some examples to them. There are, as I said, many gaps in rational drug discovery, but I think one of the main gaps is the consideration of mutations in every very early stages. I think this is perhaps changing in the last couple of years because with COVID-19, people are started to be more and more exposed to the concept of mutations. So we specifically work on missense mutations. These are genetic variations that results in changes in amino acids in a protein. As I said, ribosomes produces the protein, and protein falls into three-dimensional structures. Sometimes many of them comes together and makes quaternary structure. And sometimes one single amino acid change in a protein can cause really drastic changes in the function and the structure. And the best example, I guess, is sickle cell anemia. There is only one nucleotide change in beta globin protein in sickle cell anemia, which then makes the hemoglobin not in normal form, but in long form, an inflexible chain, which then changes the structure of the red blood cells, as you can see here. I would like to present look at the missense mutations in two different aspects of drug discovery here. So first aspect, when we have disease, we try to understand the disease, of course, and we design the drugs accordingly. And if we have any protein drug target that's great. We can use it to design the drugs. But no matter what we design, especially in infectious diseases, we always get resistance, right? There is no escape from that, it seems. But this is not only for infectious diseases. We have the same problem for non-communicable diseases as well. For instance, for cancer, drug resistance is a very serious problem. And the main reason for this drug resistance is, again, mutations. The second aspect is drug metabolism. So when we are sick, we get drugs to feel better. But this sometimes backwards. I mean, we get really sick. We don't get better by taking the drugs. Why? Because we are all different, actually. Our genomes are different. We have mutations. We call them variations, not mutations in human genome. Drugs are strangers to our body. So when we take drug, it goes through number of proteins and steps and to get metabolized so that we can get them out of our body one way or other. And if we have variations in these proteins that metabolize the drugs, then those drugs might have side effects. Okay. And sometimes we get drug toxicity. So again, mutations. Okay. So for long-lasting, more effective and safe drugs, we need to understand the molecular mechanisms of these missense mutations on protein level. And these proteins can be either drug targets or pharmacogens that actually metabolize the medications that we take. But it is not a trivial job to understand the mechanism of these mutations. So let's say you have protein, and if the mutation is in the active site, functional side of the protein, then it's easier because it changes the interaction between the drug and the mutation. But if the mutation is somewhere else, and normally it is most of the time the mutations are not in the functional site because pathogens actually want to use that protein, not to kill their own protein. So they actually cleverly mutate in a way that these mutations affect the active site when it's necessary, when there is drug in the environment. So there is a long communication path here. And sometimes many of them comes together to create this drug resistance. Okay. And we call them allosteric communications, and these mutations are the allosteric mutations. So allosteric communication is like Chinese whisper to me. So you give an input, and these are, let's say, individual amino acids in the protein, and this input comes back in the active site. Okay. What happens if there is a person with hearing impairment in between? The communication changes, the message changes, and allosteric mutations are like this hearing impairment. Okay. We started to work on mutations roughly 10 years ago under the H3 AbioNet, which was a bioinformatics network for H3 Africa. And H3 Africa aimed to get lots of human genomes out, African human genomes, from healthy people and the people with specific diseases. And the idea was then you can compare and see which variations are the ones causing these mutations. We had no interest to genome analysis that time, genome sequencing, neither now, because I think the whole research for me starts after getting the information from genome. What do you do with those variations? How do you analyze them? How do you link them to diseases? Because getting the variations is the first step. And we were the only group in the network decided to look at the single nucleotide polymorphisms or missense mutations on proteins. Just to give you an idea about the amount of mutations that we have in Africa. African genomes are very diverse, and there are lots of variations compared to other populations, other continents. Okay, and it tells here that 3.1 million single nucleotide variations from one data set, but actually total they looked at number of populations in Africa, as you can see here. And altogether, they identified 41 million high-quality single nucleotide variations. So actually, we have more than that in human genome, roughly 900 million variations. I had to think a bit. Okay, so how do you look at them? I mean, there's no way that you can do clinical tests or wet lab experiments to analyze all these genomes. With that in mind, we thought that we actually can establish some computational techniques to screen them. And I think this is one of the best contributions that we have to the field. We establish dynamic residue networks. Here, we think that each residue connect to other one in protein, and they communicate, and we carry that over the molecular dynamics simulations, where we mimic basically how the proteins would behave in cell-like environments. This study started with David Brown. He was my software developer, but also PhD student, and continued with some other people, and David Penkler, Olivier, and so on and so forth. Okay, so very briefly, I would like to talk about how we applied our computational approaches to different aspects of drug discovery. One aspect is, of course, analyzing the current drugs and understanding the resistant mechanisms, which would help us then to look at, to develop the drugs further to perhaps circumvent the resistance mechanisms. This is one of our very first studies. This is Olivier's PhD project. And here we looked at 1,600 structures, HIV structures, and we were trying to understand if there is any mechanisms. I mean, because in HIV, whatever drug you have, you get drug resistance. HIV protease is one of the key drug targets, and all the drugs are binding to active site. What we found, it doesn't matter what drug binds to the HIV protease, the active site, there is a common actually behavior due to the mutations, and that is there is lateral expansion. And you can see this represents the behavior of the protein in different drugs. But generally, there is lateral expansion, as I show here, and there's contraction here. So it expands like that and contract from the bottom. And we claim that, we hypothesize that then dislocates the drug. So perhaps the better way of designing drugs for HIV protease is to use other sites rather than active site. We also looked at TV drug resistance. Here we worked on pyrazinamide, PZA, and 83 different resistance mutations, and we realized that in certain mutations, drug just comes out of the target, the protein. And again, we identified a common mechanism for that. That was the opening of this loop region here. We also looked at rifampicin, which is a well-known TB drug, and there is a resistance to that, as well as isoniazid drug resistance. In all of the cases, I mean, these are very specific cases, and every case is different actually. And the protein, as protein changes, drug resistance mechanism changes, it seems. We have little bit studies in malaria, but it's harder to work in malaria because there are not much known protein drug targets. Yeah, we have the drugs, malarial drugs, but mostly don't know where they are binding actually. Okay, this work was not, most of the work was possible because of this grant, and I had really great pleasure to work with senior program officers from Bill Gates, MNV, and the scientists with H3D, as well as the people grantees together. It was very nice support for us. The other question, of course, that we need to ask, how do we design drugs, inhibitors, if the protein is continually evolving? I mean, this is totally ignored. Any drug is designed for just one thing, but these pathogens are actually evolving. So of course, this brings us to COVID. This is the best example, right? And we did a number of work here. We have 2 publications came, and in each case, in these publications, we look at main protease as drug target. It doesn't matter what this shows. What I'm trying to show here, actually, there are different calculations, and this is the protein from Wuhan strain. Okay, Wuhan strain and Wuhan strain. Look at how the behavior of protein is changing in the presence of mutations. So some of the mutations are really changing the function and structure of the protein for good or for bad. Okay. So this led us to a new grant actually, and I'm very excited to get this grant as part of PED initiative from Novo Nordisk Foundation. And I'll be working with 2 colleagues from University of Cambridge, Stephen and Ian, and I'm looking forward to start this grant. And I would like to specifically thank Rob, Jordan, and Tina and Fiona for their continual support in this grant. Now, in the last few minutes of my talk, I would like to stop and take my time in pharmacogenomics. I think pharmacogenomics is extremely important in the context of Africa. Let me first define pharmacogenomics. It analyzes how the genetic makeup of a patient affects his or her response to drugs. I talked a little bit about this. I mean, we are not same in our genome, so not every drug is good for everybody. So Africa is home to some of the most genetically diverse populations. There are roughly over 3,000 distinct ethnic groups and wide range of variety. And Africa also has lots of diseases, and each of diseases requires, of course, treatment. But because there is so much genetic variation, treatment has problems. It doesn't fit to all of us. And more than that, actually, there is only very little pharmacogenetic research has been conducted in Africa. So this brings me to our project. I have a large team here. We published our first article this year. What we are trying to do, we are trying to set up a kind of common approach, a computational approach to predict if the malarial and TB drugs can be effective or toxic in the presence of certain variations. And this is supported by Project Africa Gradient. I'm so privileged to work with many people in the Gradient. We call it Gradient family, as well as these Innovatis and GSK and SAMRC people in this project. This was announced so broadly actually and publicized so well everywhere, and how Novartis and GSK decided to actually collaborate to support the pharmacogenomics in Africa. I'm almost at the end of my talk, and I thought I have to acknowledge all my students. I will not be able to talk about their work, but I have also a team of people works in tRNA synthetases, and we think that this is another way of PAN inhibitors is another way of actually trying to win against drug resistance. I have one student in microsporidia and genomics work, and I have to thank Stacy specifically for being so helpful in all sorts of admin work to me. The other collaborators that I haven't mentioned, past and current, are listed here. I hope I haven't forgotten anybody. And the funders, I mentioned a number of them. I specifically would like to thank CHPC here. Without them, we couldn't do any of our research. And of course, my family, my mom, my late father, my brother, his family, my husband, and my child, who is almost sleeping. Thank you very much. Okay. Thank you. Teşekkür ederim.
Thank you. Wow. Özlem, we always get to see your name a lot at Grad. We get to see all the papers, we get to see the awards, and now we got to see you. Thank you. This is a public lecture where you're introducing yourself to the community, and we've got to see the humanity that's in you, the care that you have with your students, the team that you've built up. You make us proud. Thank you very much. And this is a little token from the university, from us. But most importantly, your students have asked to come down and present with you something, which is an absolute testament to the team that you've built up, the relationship you have with your postgraduate students, and the fantastic fun research it's doing. So thank you for inviting us into this world of bioinformatics, this beautiful synergy of looking at computational chemistry with biochemistry and microbiology. I think we've got a little bit of better understanding what you do. It was fantastic, thank you. And ladies and gentlemen, I'd like to invite you to come outside. We have some refreshments and some nibbles, and we can celebrate. Yeah, Özlem, you're a professor. Well done. Thank you.